About HLA and kidney transplantation. With respect to major histocompatibility complex HLA, what are class 1, class 2, why this important in kidney transplantation, that will be covered in a separate topic. In this we will see how the HLA reports are being interpreted for the purpose of kidney transplantation. For kidney transplantation, the important HLA are HLA, A, B and DR. This is the major important HLA which are being considered for kidney transplantation. Suppose if this is being analyzed for the purpose of kidney transplantation, this is called as low resolution HLA typing. Suppose if we see the entire HLA 1 and 2 and other allele also, that is HLA A, B, C, DR, DP, DQ, that is called as high resolution HLA typing. There are two types of HLA typing. Before going into the examples and how to match it for kidney transplantation, first we will see about the nomenclature of this HLA. HLA is being denoted as HLA followed by a separator and denotes the gene. Then there is another separator. The first will tells about the allele group, second will tells about the specific HLA protein, third about the DNA substitution, fourth about the changes in the non-coding region, and the suffix tell about the changes in the expression. So we are having field 1, field 2, field 3, field 4. In the low resolution typing, we will be having only up to the field 1. So in the high resolution typing, we will be having up to field number 3. So HLA nomenclature with allele resolution and 4 digit, that is 4 field up to 8 digit. With respect to the last prefix, it can be low cell surface expression denoted as L, soluble secreted molecule denoted as S. If it is in the cytoplasm denoted as C, A is aberrant expression, C is questionable. Not expressed as null alleles are denoted as SN. This is a summary about how the HLA being denoted, HLA protein, specific HLA protein, how we can denote it and what are the individual letter denotes. So first we will see about the one example of low resolution HLA typing. Here the patient and the donor details are given. This HLA A1 and A11 of the patient. This is about the donor. How this can be denoted? I am writing about only the HLA EA24, HLA EA31. Similarly, for the recipient, it can be denoted as HLA A01, HLA A11. So this is the HLA A loci for the recipient and the donor. So basically this is what the number denotes here. So we will be matching it with the donor and the recipient. Here 1 and 24 not matching, 11 and 31 not matching, 18, 40, 52, 57 not matching, 7, 4, 15, 10 it is not matching. So there is 0 by 6 match. Out of the 6 allele, that is A, B and DR, we have seen none of the alleles are matching. So they have mentioned that the report is being given as the patient so and so / no common antigen at HLA loci 1 or 2 with respect to the donor. They have did the CDC also. Regarding the CDC and flow cross match, we will see in a separate video. But with respect to the HLA, there is, in this example, there is 0 by 6 match between the recipient and the donor. This is the low resolution example of low resolution HLA typing. The report was being from names. Then we will move to the high resolution typing. This was high resolution typing, that is the next generation sequencing. We are having up to 3 field as I shown in the example. So how this, for example, how this field can be, this is for the patient, this is for the donor, this is the recipient, and this is the donor. So how to write the for example, I am writing the HLA allele for the HLA-A loci. So it is written as 01 01 01, and HLA-A loci, another location is 31 01 and 02. Similarly, if we write for the donor, it can be written as HLA-A asterisk 03 01 01. For the HLA another loci 31 01 02. So this is how this A loci, how it is being written, and how it is being written for the A loci complete. If we want to write, it will come like this. So now we will go for the matching. If we take 31 1 2 matching, this is matching with this one. 3 1 1, it is 1 1 not matching with respect to the B loci. 35 3 1, it is matching with 35 3 1. Coming to the C loci, it is 12 3 1. 12 3 1 is matching. Coming to the DP1, 2 1 2 2 1 2. Coming to the DQ, 5 1 1 5 3 1 5 3 1 5 3 1 is matching. 15 14 51 1, this is matching. So if we calculate, so if we calculate 1 2 3 4 5 6, and one more is also matching, this one 4 2 4 2 1. So out of 12 we are having 7 matches. So this is being given as 7 out of 12 match in this high resolution. This is what they have given as the result over here. So once we come to the result, so here they have mentioned as the patient so and so is 7/12 match at allele with the donor at HLA class 1 and class 2, that is the DP, DQ and DR. So this is the result of the high resolution HLA typing. Usually mismatch is more important than matches within kidney donation. So usually, but they give the result as how much mismatch. If a patient, if between HLA between a father and mother or a parents and sibling, usually always half match. If it is 3/6 or 6/12 in a low resolution in high resolution HLA typing, this is called as being called as haploidentical. Why it is coming like this, that we will cover in the basics of the HLA. So these are all the 2 examples. We will just quickly go through the another 2 example, one for low resolution and one for high resolution. Just we will focus over here. So how to interpret this? This is the HLA type of a patient and the donor. Donor is the wife. So we will quickly go for the matching. Patient not matching, 33 68 not matching, 47 27 not matching, 58 40 not matching, 13 is matching. So you can say the result is being given as 1/6 match in this low resolution type. Leave the CDC. This is the extra report that is being given that I will cover in a separate video. So once you go to the remark, the patient so and so shares 1 out of 6 antigen at class 1 and 2. This is low resolution typing that we have seen on HLA A, B and DR. So 1 out of 6 match. Coming to the another example for high resolution, quickly going through. So this is the recipient, this is the donor. So we can start matching. 24 to 1 is matching, 24 to 1, 2752 matching, 7 to 1 matching, 411 matching, 311 matching, 1111 matching. So 1, 2, 3, 4, 5 and 6. So the result is, out of the 12 are matching, 6 are matching out of 12. In this high resolution typing, as we see here, the result is given as same matching ratios patient 6 by 12. The patient so and so having 6 by 12 match at allele level, so they are almost half match. Probably the recipient and the donor might be the parent and offspring. So this is about high resolution HLA typing. This is about low and high resolution HLA typing with respect to kidney transplantation and how to interpret the report. Regarding the advantage and disadvantage of this low and high resolution HLA typing and the basics of the HLA and MSc, we will see in a separate video.